How would you solve a problem if you needed to know the probability of two different traits occurring at the same time? Your answer? A dihybrid cross. Today we're going to be talking about dihybrid crosses, which is when we do a Punnett square, but we're going to be looking at two different traits. So questions that could be asked are things like, what's the probability of producing a yellow flower if two heterozygous plants are crossed? And what's the probability of producing a round seed if two heterozygous round plants are crossed? So we want to know what's the probability of having round-seeded yellow plants, for example. So a cross that deals with these traits is called a dihybrid cross. They can be solved with a larger 16 square Punnett square. It can be smaller if you don't have that many alleles available to you. So like if say you have a homozygous dominant individual for both traits, you only have to do kind of two squares along that line because there's only two options for allele combinations. Now, Mendel's law of independent assortment was created from a study of dihybrid crosses, and it states that genes assort independently. So one dominant gene or one recessive gene does not affect the location of another one. So Mendel cross-pollinated pure breeding plants that produce yellow rounded seeds with pure breeding plants that produce green wrinkled seeds. So pure breeding round were capital R, pure breeding wrinkled were lowercase r. Pure yellow, capital Y, pure green, lowercase y. So the genotypes of these plants would have been capital Y, and capital Y, lowercase r. Now, the F1 generation of these plants are heterozygous for both yellow and round genotypes. And in a dihybrid cross, there are 4 possible phenotypes. So we want to cross a heterozygous for 2 trait plant. So when we're looking at our parent genotype possibilities, we have the possibility type of having a capital Y, capital R, capital Y, lowercase r, lowercase y, capital R, lowercase r for both individuals. So when we put in our, I'm not sure if I'm going to have enough room here, 16 block Punnett square. Those are going to be pretty small. We have YR, 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 YR. And same for the next one. So we would have YY, RR, capital R, lowercase r. Oh, that's a lowercase y. R, 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 R. It's 2 capital R's there. 2 capital R's, and then this was a lowercase r, not a y. Okay. We would have capital Y, big R, little r, capital Y, little r, capital Y, lowercase y, big R, little r, capital Y, lowercase y, little r. Okay, and I always like to put my capital letters first, so capital Y, lowercase y, big R, capital Y, lowercase y, big R, little r, lowercase y, big R, lowercase y, big R, little r. Okay, and the last one I'm kind of just going to overlap here. Capital Y, lowercase y, big R, little r, capital Y, lowercase y, little r, lowercase y, big R, little r, and then lowercase y, little r. Okay, fit it all in. So it takes a lot. Once it's not so messy on the screen, you can see what you end up having. But here we have our one homozygous dominant individual. Down here we have our one completely homozygous recessive individual, and then we have a combination of other ones in between. So we end up with a really nice ratio for these heterozygous crosses. So the probability of all the chances and the total number of combinations is 16 in a dihybrid cross. And the rule of independent events means that previous events do not affect future events. If you toss a heads and a coin, the chance of you tossing heads again is still 50%, no matter what's happened before. And the product rule states that the independent events occurring simultaneously is equal to the product of these events occurring separately. So the chance of tossing heads after 2 tosses is 1 out of 2, but the chance of tossing heads 3 times in a row is 1 in 8. So it's 1 in 2 times 1 in 2 times 1 in 2. So you can calculate dihybrids by using 2 monohybrids and using the product rule. So if we look at the chance of getting a homozygous dominant, and homozygous dominant is 1 in 4. We multiply them together, that's 1 in 16. Or a heterozygous is 2 in 4, or 1 in 2. And a heterozygous is 1 in 2. So the chance of that would be also 1 in 4. So here is a much nicer description of the the dihybrid cross that I really disgustingly drew out. So we have our 2 homozygous dominant and homozygous recessive at 1 in 16 each. We have our 4 heterozygotes in here. So 1, nicely in the middle, 2, 3, 4, for a chance of 1 in 4 having heterozygotes. And then the rest, we have a variety. Here we have our yellow plants, so everything above kind of this line is going to be a yellow plant. And then everything above then we'll have our round ones kind of mixed in as well. So here is our different ratios. So we have, everything needs to add up to 100. We have 6.25% or 1 in 16 homozygous dominant. We have 2 in 16 that are yellow and round. We have 1 that's yellow and wrinkled. We have, here's a genotype, so another yellow and round, yellow and round. And then we have yellow and wrinkled, green and round, green and round. And there should be a lowercase y here. And that's our green and wrinkled plant. So everything there adds up to 100%. And all those fractions should equal 16 out of 16. So for our dihybrid crosses, you must identify what the dihybrid stands for. So for our genotype here, we have to give our different allele combinations, which one is dominant and recessive. And you don't often have to do genotype ratios for dihybrid crosses. But you see there, that all adds up to 16. And there's our common heterozygote for both traits in the middle. But for the phenotypic ratio for a dihybrid cross, this is our common phenotypic ratio, which is 9. Show the dominant traits. 3 will show 1 dominant, 1 recessive. 3 will show the other dominant, another recessive, and 1 will show both recessive traits. So a dihybrid cross of 2 parents heterozygous for both traits will always give a 9:3:3:1 phenotypic ratio. And so in the last one, this would have been, where's my pen? Would have had 9 that were yellow and round, 3 that were yellow and wrinkled, 3 that were green and round, and 1 that was green and wrinkled. So that's our phenotypic ratio for a heterozygous dihybrid cross. So when you're doing your dihybrid cross, you can identify which gametes are going to be possible. So in this case, we have a dominant for B and dominant for R option, or recessive for B and dominant for R option. So there's only 2 options for gamete combinations. For our second individual, we have dominant for B and lowercase for R. So incidentally, only 1 gamete option for that individual. So again, what's the probability of producing a tall red flowering plant if 2 plants are crossed. So we're going to have tall and red, tall and recessive. Oops, sorry. Okay. And we should be able to do this with the Punnett square, and then we should also be able to just do it with our ratio. Okay, so we want a tall red flower plant if two plants heterozygous for both traits are crossed. So it actually should be about 1 in 16 to produce a tall red flowering plant. Or sorry, it should be 9 in 16. So when we go through, so that's our heterozygous dominant. This one's also tall and red flowering. So is that one. And so is that one. This one is as well. This one is not. That one is. And this one is not. That one is. That one is. This one is not. And that one is not. Oops, I kind of messed up there. But that one is tall and red flowering. It's a capital T. So that one's not. This one is not. And this one is not. So we can go 1, 2, 3, 4, 5, 6, 7, 8, 9. So we had our 9 out of 16 were tall and red flowering plants when two heterozygous plants were crossed. So that's the dihybrid cross Punnett squares. They're a lot larger. They take some more time to figure out your exact genotypes in the offspring, but they are great for solving those problems. Take it easy. Bye.